Hi, everyone, and thanks for joining this Southern Research webinar. My name is Kaylee Minkiewicz, and I'll be the moderator today. In this webinar, Dr. Joseph Randall, the Director of Toxicology at Southern Research, will present his talk titled Neurovirulence Testing of Live Viral Vaccines. During this presentation, if you have any questions, you can submit them using the Q&A button at the bottom of your screen. Please put your questions here, not the chat window, so we can be sure to record your questions to answer during the Q&A session following the presentation. So without further delay, I'll turn this time over to Dr. Joseph Randall.
Hello, it's a pleasure to speak with you today. So what I'd like to talk to you today is about the central nervous system safety testing of live attenuated viral vaccines. The CNS, the safety of vaccines has received a lot of public attention since the SARS-CoV-2 pandemic, and we have invested in new technologies to evaluate the CNS safety of vaccines. So today I'll be discussing the neurovirulence test, which is designed to evaluate whether vaccines are sufficiently attenuated to remove central nervous system side effects. So that's the major focus of today's talk. Discussing 4 items today: the monkey neurovirulence test validation using yellow fever vaccines, as per the WHO guidelines, how we've refined our stereotaxic surgical methods for intrathalamic drug delivery of live viral vaccines into the brain, as per the European Pharmacopoeia guidelines. And we've developed a mouse model of neurovirulence based on intracranial dosing of live yellow fever vaccines to confirm neurotropism as reported in the literature. So this mouse model is an adjunctive model and is much more sensitive than the monkey neurovirulence test, which is the gold standard model for evaluation of oral polio vaccines. And this monkey neurovirulence test has been used for over 75 years. The 4th objective is to provide a better understanding of the predictive value of rodent neurovirulence models when compared with the gold standard monkey neurovirulence test. At Southern Research, we previously conducted 3 neurovirulence studies in cynomolgus monkeys. 2 studies were conducted with dengue fever master seed vaccines, and we're not going to be discussing that today. I will be discussing 1 study with yellow fever master and working seed vaccines that were tested in comparison with a commercially available yellow fever vaccine, Stamaril, and the WHO reference vaccine, 17D 16873, which is a live attenuated virus. This information is from the Tyagi et al. Publication from 2023. And top-line results were after intracerebral dosing of yellow fever vaccines, all the monkeys survived, and there were no adverse clinical effects or signs of encephalitis or paresis. So the results of the test were that none of the test vaccines were neurotropic in the neurovirulence assay. But I'm going to go over some of the details of that study, which included endpoints for clinical activity scores and histopathology of the brain and spinal cord. We also measured viral titers in the blood and anti-yellow fever antibodies using a PRNT assay. The monkey neurovirulence study had 6 treatment groups. There were 3 test vaccines: the master seed virus, the working seed virus, and the commercial vaccine product, P240. The comparators were Stamaril and the WHO reference vaccine and a placebo control. So there were 6 groups with 5 animals per sex per group, and the dose volume that was injected into the brain was 1/4 of a milliliter. And all of the animals survived and had no deleterious effects after intracerebral administration of the vaccines. And blood was collected for viral titer by plaque assay on days 0, 2, 4, and 6. And blood was collected for immunogenicity and the determination of neutralizing antibodies to yellow fever in serum. And finally, the brain and spinal cord were collected at necropsy and evaluated histologically. So this is a summary of the yellow fever neutralizing antibody results in serum. And you can see here that this is the plaque reduction neutralization titer, where you see a 50% reduction in the viral plaques when you add serum from the vaccine-treated animals. So animals that received the master seed virus, the working seed virus, or the final product all had high levels of neutralizing antibodies, as did the commercially available yellow fever vaccine, Stamaril, and the WHO reference vaccine. The only animals that did not have neutralizing antibodies for yellow fever were, of course, the placebo control, because they were not exposed to any of the vaccines. This is a summary of the mean clinical scores for the different treatment groups, and also the histopathology scores as well. Side by side, you can see on the left is the treatment, the master seed virus, working seed virus, the final product, Stamaril, and the WHO, and the placebo. All of the groups were compared statistically against the WHO reference vaccine. P-values are reported and significance at the P less than 0.05 level. And the clinical grading score was on a 5-point scale, 0 to 4. So the mean clinical scores were less than 1 for all of the vaccine test groups. And they were comparable or not significantly different than the WHO reference vaccine, which had a mean clinical score of 0.24 on that 4-point scale. Stamaril is the only treatment that had higher clinical score relative to the reference vaccine. And you can see this with the clinical scores. Were about double the WHO reference vaccine, as were the histopathology scores. So if you look at the histopathology scores, discriminator areas with different regions of the brain, the average scores was less than 1, which would be mild findings. And Stamaril had a score of 1.19 relative to the WHO reference vaccine, which was significant at 0.001 level. And another way of calculating the histopathology scores, you also see that Stamaril had a slightly higher histopathology scores relative to the WHO reference vaccine, and then it was significant. However, none of these animals developed paresis, encephalitis, or any neurological effects. So getting to the conclusions of this validation study, no animals developed signs of encephalitis. No animals had paresis or tremors after intracerebral vaccination. The vaccines and placebo were well tolerated, and no significant body weight loss or adverse clinical signs were noted in these animals. The mean clinical and histopathology scores in monkeys dosed with the 3 working viruses, the test viruses, were not significantly different than the WHO reference vaccine. And that means that these 3 vaccines passed the neurovirulence test and were negative in this test. So the serum load was measured in terms of the infectious titer in the blood, and there was detectable levels of yellow fever in the blood on days 2 and 4, but by day 6, none of the animals had detectable viral titers. So the virus had been effectively cleared from the blood after 6 days of vaccination. And as I presented previously, there was 100% seroconversion for the animals that were treated with the yellow fever vaccines, and viral titers antiviral titers were relatively high on days 14 and 30, with the exception of the placebo control. Stamaril had higher clinical and histopathology scored versus the reference vaccine, but this was not considered to be biologically significant since none of the animals had clinical signs of neurological impairment. So that's a summary of the results from this Tyagi et al. Paper, and additional details can be found therein. So we were recently faced with a challenge where a European client wanted to administer the drug, the test vaccines directly into the brain, but in compliance with the European Pharmacopoeia guidance. The European Pharmacopoeia guidance is different than the WHO monkey neurovirulence test in that the vaccines are supposed to be delivered directly into the brain, into the thalamus. And you can see here in the right-hand figure that the vaccine is administered into the left and right hemisphere, and the orange highlighted area is the thalamus. So it's in the middle part of the brain, and it is involved in the regulation of consciousness, sleep, and alertness. So lesions of this area would cause activity changes and or neurological symptoms. The European Pharmacopoeia test is simply a single injection on day 1 on both left and right thalamus, and to observe the animals for 21 days, followed by evaluation of brain histopathology. However, it was challenging to determine the coordinates for the thalamus. So based on this publication here, you can see that the coordinates that we found for the thalamus, the x-axis is the medial lateral, which is on either side of the middle part of the skull, left or right. And the y-axis is rostrocaudal, towards the nose or towards the back of the head. And the dorsoventral z-axis is the depth that the needle has to go to reach the target area. And so we looked up several stereotaxic atlases of rhesus monkeys, and we tried those coordinates in cynomolgus monkeys, and they were not accurate. So what we did is that we placed the animals into a stereotaxic chamber, and the center point was in the middle of the skull was relative to the bregma. So we empirically determined the accurate coordinates for the thalamus by injecting trypan blue dye into the brain, and then macroscopically evaluating where the needle was placed and whether or not we had proper drug delivery in order to meet the European Pharmacopoeia guidance. So the coordinates that we have determined empirically is on either side of the midline, plus 4.5 or minus 4.5 millimeters, minus 2 millimeters basically towards the back of the head. It's basically right at the bregma, and 22 millimeters in depth. Those are the coordinates we determined for the thalamus in cynomolgus monkeys. So this is the study design that we came up with for the European Pharmacopoeia neurovirulence study. And we had basically 2 treatment groups. We had 5 males and 5 females were treated with the live vaccine. The dose volume was 1/4 of a milliliter injected into the thalamus, left and right hemisphere. On day 1, or day 2, we staggered the dosing over 2 days. And then there was a satellite control group that did not receive surgery or sham vaccination. So those animals were followed until day 32 or day 33 as per guideline. And the animals were anesthetized and received live viral vaccines and recovered well after the surgery. We evaluated immunogenicity by measuring antibody titers by ELISA prior to dosing and at study termination. And we measured the following study endpoints, clinical observations, body weights, food consumption, all of that. And the brain was collected at scheduled necropsy, as was the cervical and lumbar spinal cord, and they were processed and evaluated to slide to look for basically lesions in central nervous system. So one of the disadvantages of this design is that there is no placebo control, and it's not required by guideline. So what we noticed in the first study that we ran with this guideline is that we had needle track findings in half of the animals. So in the thalamus, we saw mild to moderate inflammatory changes or inflammatory infiltrates in the thalamus of these animals, and they were associated with the needle track lesions. Of course, these findings were not observed in the satellite controls because they were not injected with placebo-controlled material, and they did not have the surgical procedure. So we recommend caution when conducting studies of this design because it's very difficult to rule out a possible effect of the vaccine. And in this case, I believe that for at least half the animals, the mild to moderate inflammatory changes in the thalamus were due to the dosing procedure and not necessarily the vaccine. So I wanted to change gears here and let you know that there is a severe shortage of monkeys for biomedical research. And in fact, the monkey neurovirulence model, while it is the gold standard for oral polio vaccines, is not considered to be sensitive to all different viruses. So in other words, no single neurovirulence model is considered appropriate for all vaccines to be tested for neurovirulence. And so we've endeavored to develop a neonatal mouse model of neurovirulence, and we ran a pilot study as described here. We had 4 treatment groups. We ordered pregnant dams, and we administered the drug. Intracranial administration of vaccine was on postnatal day 4 to these suckling mice. And daily observations were reported beginning on postnatal day 1 and for 11 to 13 days after that to determine neurovirulence as measured by morbidity or mortality, body weight changes, and clinical evidence of neurological impairment. So the group 1 animals received 20 microliters of phosphate buffered saline. Group 2 animals received 20 microliters of the WHO reference vaccine at 50 PFU, plaque forming units, and group 3 received a trypan blue dye. Group 4 was untreated. So basically, this is a screening neurovirulence test. Offers a lot of advantages over the monkey neurovirulence test. So the results from this study were as follows. The survival endpoints were met. We had only one of the control pups was lost after transient hypothermia-induced anesthesia on postnatal day 4. All the other animals, all the other mouse pups in the PBS control, trypan blue, and satellite control groups survived until scheduled euthanasia. Intracranial administration of the WHO reference vaccine caused hyperactivity or hypoactivity in mouse pups after only 6 days of dosing. and the pup stopped weaning on day 7 and had to be euthanized due to hypoactivity and to alleviate distress. These results indicate that suckling mice are unusually sensitive to the WHO vaccine. And I want to reiterate that this is a live attenuated virus, and the dose was only 50 PFU. And we saw 100% morbidity and had to euthanize the animals in group 2. In a follow-up study, intracranial administration of 100 PFU of commercial yellow fever vaccine induced hyperactivity or hypoactivity in all mouse pups were euthanized by postnatal day 11, which is 7 days post-dose. So a lower dose of Stamaril was better tolerated in suckling mice, and we saw greater than 75% survival for the 3-week observation period. So this is the survival curves looking at viability, percent viability over time, and postnatal day 5 through 25. The animals were received a single intracranial injection on day 5, and they were monitored for a 21-day observation period. You can see here that the animals that received 100 PFU died before day 15. Actually, they were taken down before day 15 due to side effects. And at the mid dose of 10 PFUs of the Stamaril vaccine, there was greater than 75% survival. So there's a clear dose response relationship here. You can see there was 100% survival in the animals that received 1 PFU of the vaccine or less. So this is a good comparator, in my opinion. So one of the advantages of conducting a study like this is that you have greater statistical power. You can evaluate the dose response, and you want to pick a dose that caused 50% loss of the mice due to the neurotropic effects of the vaccine. So in conclusion, no single neurovirulence model is appropriate for testing of all live viral vaccines for neurotropism. For example, the monkey neurovirulence test is not appropriate for evaluation of mumps vaccine safety. And there's ongoing work by Steve Rubin and others to develop a neonatal rat model, which is more predictive of effects in humans. And in fact, the mumps vaccine is not required. It's not required in Japan because the vaccine that was developed for that market failed in the monkey neurovirulence test. So what happened was they developed a vaccine. It was tested in monkeys and was considered to be safe. But when it was released to the market, that vaccine produced deleterious side effects at an unacceptable rate, and that product was removed from the market. So the neurovirulence testing is critically important to protect patients from potential neurological side effects of vaccines. So Southern Research supports the 3R principles, including the refined use of lower animal species for non-clinical testing. So we're very much interested in further development of the yellow fever neurotropism test in suckling mice. And we actually have 2 clients that are working with us now on this neonatal mouse model. So rodent models are robust, in my opinion, and allow for comparison of novel and approved vaccines for neurotropism or neurovirulence, because you can have a larger size treatment groups. So in the monkeys that are so scarce, we barely have enough animals to run 5 animals, 5 males and 5 females in a group. And we certainly don't have enough to run many groups. So we often test only one concentration of the vaccine and not multiple doses of the vaccine. So the advantages of the rodent models, you can test, 5 different dose levels. You can have greater statistical power. The test is faster and it's easier, but it's not currently accepted as an alternative to the monkey neurovirulence test. So it would be an adjunctive model for our clients to present data to the FDA. So I mentioned before that 50 PFUs of the WHO reference virus and 100 PFUs of Stamaril are neurotropic in mice, in neonatal mice, and these doses are ostensibly not neurotropic in monkeys. And that's because these live attenuated viruses, at least the Stamaril, is commercially available and it's not associated with neurological side effects in humans or in monkeys. So the mouse neurovirulence test is a lot more sensitive, and you can evaluate the dose response relationship of test vaccines versus commercially available vaccines. And the final comment is that the validation of rodent models of neurovirulence requires industry and regulatory collaboration. For example, there is a transgenic knock-in mouse model that is used for neurovirulence testing of oral polio vaccines. And that took many years, but it is available. So I think that we need to, as an industry, invest more in these alternate models to understand the pathogenesis, and the mechanism of attenuation is poorly understood. So there's a long way to go in developing additional animal models, but this is just some small steps that we've taken at Southern Research to promote vaccine safety testing and development. Thank you very much for your time. I have a few references here with regards to the papers and the test guidelines, and I encourage you to take a look at those. And I'd like to thank you very much for your time and to ask you if you have any questions.
So now we will begin the Q&A portion. Again, if any more questions come up, please submit them using the Q&A tab. Dr. Randall?
So with the first question, what is the source of the target virus being used in the PRNT assays? I would have to confirm with my colleagues, but it is likely the WHO reference virus. But I would have to confirm that. Additional information is published in the paper. So one of the other questions in the monkey neurovirulence test is the brain regions examined as a coronal section or separate sections for the left and right areas. So in the standard monkey neurovirulence test, I think the coronal sections were used, and we didn't specifically section the left and right hemispheres. We just used our standard operating procedures for brain sections, but we did score for the purposes of the European Pharmacopoeia study, we did score the left and right hemispheres separately. They both received an injection. So how well established is the mouse neurovirulence test as compared to the monkey model for other vaccines? And these data show that the mouse model might be too sensitive. And I would agree with you that the neonatal mouse model is overly sensitive, and you would have to take the data into context. So it's not currently a replacement for the monkey neurovirulence test, our clients are using the mouse test as an adjunct to provide additional information on the dose response for neurovirulence in the mouse model. So some of our clients have tested from 0.01 PFUs all the way up to 1,000 plaque-forming units. So a very, very wide range of virus has been used in these models. And in comparison, instead of using wild-type viruses, which are more dangerous and harder to manipulate, the positive control for this mouse neurovirulence test can be Stamaril or the WHO reference vaccine. And so it's easier to use those vaccines, but I think data needs to be taken in context. There was a question about how the mouse models of poliovirus neurovirulence are regulated, and the answer is I don't know. I have not run those studies or submitted any of this data to a regulatory agency. So this information was in one of the publications, and apparently this model's been around for more than a decade. So I encourage you to look into this further, and I will look for any regulatory guidance related to the polio neurovirulence models, and I will provide that after this presentation. Thanks for your question. So is your WHO yellow fever reference? I don't believe it's not the 99616. It's a different strain. We may have that strain of the yellow fever reference vaccine, but we used a different one, as indicated in the presentation. So one of the questions was, what was the age of the monkeys used in the monkey neurovirulence test here and in the published study? And I can just say that we used 2-year-old monkeys in general, and they were about 2 to 3 kilos. So unfortunately, it's so hard to get animals these days that they're getting younger and younger and younger. So we usually prefer to have 2 to 3-year-old monkeys or 2 to 4-year-old monkeys. They're more mature, but they're harder to get these days. So the stereotaxic coordinates that we determined for this study were based on a 2 to 3 kilo monkeys. And one of the references, the XYZ coordinates and that paper that's referenced, those coordinates will change depending upon the size of the monkey. So your question is a very good one. Thank you. So here's another question is, what is the Stamaril LD50 in the neonatal mouse model in your experiment? And we are doing some additional work to confirm the LD50, but it's going to be between 1 and 10 PFUs. And we will determine that in future studies. But on a side note, I heard from a colleague that it was ostensibly in the 5 PFU range, but we'll need to confirm that. So I did want to answer this one. Is your brain sectioning SOP available for other researchers? The answer to that question is no, but I do recommend that all of this information in terms of which parts of the brain, the sectioning and the scoring methods are all available in the WHO Technical Report Series 978, Annex 2, and Annex 5. I think it's Annex 5 where it lists the different regions of the brains that have to be sectioned, how they're scored, and how you get a composite score, at least for evaluation of neurovirulence of yellow fever vaccines. So I encourage you to look at that WHO reference guidance. With regards to the needle placement and the stereotaxic coordinates, We do not have an MRI device that would be suitable for animals of this size, so that we didn't use an MRI for needle placement. We found that the needle placement was correct in all of the animals that were dosed according to the European Pharmacopoeia guidance. The MRI device would have been very helpful for determining the correct coordinates initially, because we had to do this test empirically by administering trypan blue dye directly into the brain. Thank you.
So yeah, that's all the time we have for questions now. If we didn't get to your question, or if you want any additional information, please visit southernresearch.org, or you can reach out directly at info directly to info@southernresearch.org. Thank you again, Dr. Randall and the Southern Research team. We thank you all for attending today's webinar. Please be on the lookout for the on-demand recording in your inbox in the next few days, and have a great day, everyone.